Hello, thank you to everybody who came today to join us for this talk dedicated to SPM and in particular DARTEL and its usage for VBM. Screens. DARTEL is a tool created by John Ashburner in London. The disambiguation is quite complex, as complex as the mathematical tools behind it. And this is what I am not going to discuss today because I don't understand it. But I will try to share with you practical approaches and the very basic guidelines, how to start using it, why is it useful, and what can you get at the end. VBM is voxel-based morphometry. This is an analytic approach for measurement of the differences, local differences, in the structure of the brain. Most often it is alteration in gray matter density. The practical part we will follow today is entirely based on SPM 12 guidelines, and you can find it in the PDF file, which is always there in SPM package. It is the last chapter of the practical course, and it is called Using DARTEL. This is a T1 weighted MRI image. Typically it takes 10 to 15 minutes to acquire it, and it allows you to see quite a clear separation between cerebrospinal fluid, which is dark, gray matter, which is gray, and white matter, which is light gray, and after other extracerebral structures. You can make a manual segmentation. Segmentation means a separation of a gray matter. However, it can be biased. It's always subjective, though it is still a gold standard for measurement of alterations of brain structure. But an objective measure is to make an automatic gray matter segmentation, and it is very hard. And that is why we need DARTEL. DARTEL allows an accurate inter-subject registration of brain images, which is necessary for both proper separation of different segments, different tissue classes, and after it gets partially normalized towards common global MNI, Montreal Neurological Institute template, you will be able to do group-wise statistics and to extrapolate those findings to results of other labs, other studies. This is something that might look frightening, and it is frightening. This is what is implemented in VBM, voxel-based morphometry toolbox. First, you have original images. Then, with the help of a standard canonical template, you do normalization. This is normalized image, which fits much better to the template than the original one. Then, using priors of the probability for different tissue classes, gray matter, white matter, CSF. It varies from 0 to 1. 1 is 100% like likelihood that in this point it will be gray matter. It's all probabilistic, as you see. And black means that it is never gray matter there. The same for white matter and for CSF. You see it's in the middle, ventricularly, and outside of the brain. And after this, you do segmentation of your original image. Then segmented image, which is binary. 1 means it's a tissue, 0 means there is no tissue like this. Then you do modulation. Modulation is local enhancement, suppression of the signal as a function of the deformation. If you stretch something that is originally small in order to fit the template, it will be dark. If you squeeze it, it will be bright, whiter. And then you do smoothing and then statistics. This is one picture. The next picture is even more complex. Yeah, sure. We will get there. It's more complex than, yes. It can be, but not necessary, and not always. And this is what you can see here. So this is template. This is an optimized and updated version of this toolbox, which will not run today. It's just to give you a frightening overview of how complex is the task here. First, you do registration of individual towards the template, MNI, which requires some rigid transformation. You don't make deformation, you just change the proportions. You stretch, you scale, you displace. Then you do segmentation using the priors. And then you do spatial normalization. You apply the normalization parameters to T1 image. You get normalized. Then you do another segmentation using the same priors. I remind you, it is VBM. It is not what we are going to do. And at the end, based on the parameters of normalization, the deformation nonlinear parameters, you apply local modulation, and then you smooth, and this is what you get at the end for your statistical analysis. What we are going to do is exactly like this. And this is what is recommended in SPM manual for DARTEL usage for VBM. First, reorientation, manual procedure, then segmentation, automatic. This is standard segmentation for SPM 12, which is new segment. It is already based on DARTEL. Then you run DARTEL to create template. You create a template which is local priors for tissue classes for this specific group, local group. And then with the help of this template, first you do normally, well, within this simple line, you do several different things. First, you normalize individual images towards common template, local template. Then you normalize it towards MNI template, which is easier according to the logic of the developers of this software. And then it does smoothing, and at the end we do statistics. Where can you get some data for practice? There are a few databases, and I used this one, OASIS. You can easily download their data. It is freely available, and it is mostly on aging and Alzheimer dementia. Quite an interesting project is OpenFMRI. This can be interesting not only within this task, but for other practical trials. There are a lot of fMRI databases available, but also some structural studies. And this is another one, National Alzheimer Coordinating Center. They allow you to download data samples, which is quite enough for training. If you request, then you can download huge amounts of data. Here you can download without any request. First step is reorientation. For reorientation, you use check-reg button in the SPM menu. And this is what you get. You compare two images. This is a canonical MNI normalized template, which is available in SPM. I will show you where to find it. And this is your individual image. And manually, you can apply translations and rotations. Here you have to move it a little bit on the left. Perhaps you have to bring it down. And at the end, they fit much better each other. And you apply those reorientation parameters to this image. This is the very first step. It is not totally necessary if your images are quite well aligned from the very beginning, but it is a good practice to check beforehand if all of them are aligned with each other and with the MNI. I can go to MATLAB to demonstrate this. By default, SPM locates those images not in an optimal, from my point of view, position, but you can use a very simple function in order to align them so that they cover most of your screen and that you can see all the anatomical details. Okay. Go here. We start SPM. Of course, it is tiny because it is a notebook, but luckily we can stretch this. Let's take an example from here. Those are folders with individual data samples. We have 20 subjects in total. And in each folder, we have processing subfolder where all the data already processed by me are stored. The original file looks like this, MPR something, and this is the same for all the subjects. While The canonical image is stored in SPM, and you can take, for example, simple subject T1, single subject. Now, this is what I mean. They are tiny and not quite convenient to manipulate. But if you run this little function, I will briefly open it so that you see that it is really short. Nothing complex here. You just acquire the parameters of the figure and you modify it. Let's see if it doesn't fail. Yeah, almost perfect. Now you can see that there is a totally wrong pitch here, and this is what we can correct. We reorient current image. Those are the parameters that you can set manually. For pitch, which is like nodding motion, okay. It's in radians, so it is less than 1, typically something like 0.2 or 0.3. Okay, you enter and you see that it changes, maybe 3. And after you press apply to images and it asks whether we apply it or not. We will not do it at the moment. It's just for demonstration, and I can show you the result. If you go to the same folder and you compare this one and this one, before and after reorientation, again we run this little function. This is before and this is after. Okay, this is the simplest part. And now we go to something more complex. This is what you do on your reoriented original images. And the program will automatically separate different tissue classes, different images, in fact. You can start it from here, the bottom segment on the main menu. And it gives you a batch editor. In fact, main menu is something that came from previous epochs. Actually, everything, totally everything you want to do, you can run from batch editor. And I will show you, including check range and copy, delete, everything. All the functions are available there. So what do you have to select here? Here you have to indicate that you want both native and DARTEL imported images for the first tissue and for the second tissue. What means first, second, third, and so on? It is defined in this TPM file in SPM. TPM is template, and I can show you how this template looks a bit later. First, I wanted to show the difference between native and imported image. Natives are just the same position, orientation, everything is the same as the original image. The imported is already transformed, but those are linear transformation or affine transformation. which means that you don't change the proportions. Parallel lines always stay parallel. You just stretch, you enlarge, you shrink it, but you don't apply anything nonlinear, which is typically not sufficient for proper normalization. But this is a first step to the DARTEL procedure. And this is the prior probability map for white matter. It is derived from MNI, from many subjects. Basically, it is an average of many subjects white matter, and it is smoothed. And this is what you require for proper segmentation. In fact, there are more than 3 types of the tissue. There are several extracranial tissue classes, which are also included in this part of the analysis. But we are interested mostly in first 2, and that's why we need both the native and the DARTEL important, Afinco registered images of those first 2 tissue classes. In fact, it works in a very bizarre way. First, the program is going to normalize the template towards your individual image, extract those segments, and then in the following steps after the DARTEL, there will be normalization in the other direction. So this is to illustrate the difference between the native space gray matter segment. You see it is precisely corresponds to the source image. And here we compare to registered and native. They're not the same. And here you can see why they are not the same. So here we compare the native and the registered with the gray matter template. And you can see that here there is a big gap, and here it is a little bit too much below the template, and it is moved up, and I think a little bit stretched in vertical direction, so that the difference between them is minimalized. Segmentation, because they require all those normalization steps, is an extremely long procedure. In the manual, they just stress on the DARTEL sluggishness, but in fact they are the same. So for 20 subjects, it needs an hour, and this hour means an hour on the server. It is not on personal computer. On a personal computer, it will take a few hours. Let's go to MATLAB in order to this we can close, in order to load the batch that I created before, where I can show you where to indicate all this information, which is necessary for segmentation. So it is saved batch segment. By the way, I can show also how did I create this batch segment. I didn't select the files manually. I create a file and an SPM script, which is called in my hands manager and then whatever. So we need manager segment. So it does segmentation. I indicate common path. It's very useful to separate this variable because you can do different steps of analysis on different computers. Then you check all the directories in the common path. So this is OASIS data. This is the main folder, and there are subfolder which has OAS2, like all them. So here you select this, and then in a loop you add common path, then the directory, necessary directory, and the target file. This is the source file, in fact, which will be used for segmentation. So, but the name of the file is the same for all folders. The folder, that is what is different. So you have this structure. It has to be structure of strings. So 1 by n by 20 in our case, structure containing strings. Strings, text arrays, they are used by the batch system of SPM to find the path towards the file. So then you clear MATLAB batch variable just in case. MATLAB batch, this is the structure which has a very predefined components which you can find easily in the batch editor. I will show you how. You copy this and you modify only few important lines. In this case, you modify this, and you indicate all the original files. Everything else is by default, unless you move from one computer to the other, and then you have to indicate a new location of the template files. That's it. You save batch segment, and then you can open this batch segment with batch editor. This is this file, mat file, which is read by batch editor. First, I have not to forget to show you how to extract the code. This is how to do it, quite easy. So this is all the code. You just copy paste it. It's not a lot. And then you can modify it and you can save it, which adds a lot of easiness. Yes. Sure. It's the same. The important part of my script is here, where I find and indicate the proper file for the processing. So those are the files. You can do it manually, but it is long and boring. See, different folders for different subjects. Now we don't discriminate between the groups, just all of them. And those are TPM. It is NI files, which means that it is 4D file. You have 3 dimensions, and then you have sort of different images incorporated into one file. This is a newer format than EMG, with separately a header and the main file. That's why you have to indicate 1, 2. Those are the images incorporated in this file. First, second, so on and so forth. And here you change native plus DARTEL. How do you know that this is gray and this is white? You don't know unless you go and look at TPM. Of course, I'm not going to run segment now because it is terribly long, but I want to show the TPM file. I run check reg. We go to SPM. There is a folder TPM. Those are TPM files. But like this, you select just one, the first one. You have to change something here. Then you can select 2. That's enough for us. Done. And then let's make it a bit more beautiful. Ah, I like it. So this is the template for gray matter, which is the first file, first volume of this file, and white matter, obviously, for the second file. And this is how you know what will be what in your segmentation results. Let's proceed with the presentation. Run DARTEL. This is the second longest procedure in this pipeline. What it does. It is going to use a series of rigidly aligned files, which are called RC1 something and RC2, gray and white matter. Those are individual, aligned, but still not normalized. And what is going to generate is the template. Template for gray matter, template for white matter. But those are local templates. And there will be a number of iterations in order to generate this template. It's like you normalize, you fit to each other, you average up, then you try to fit even better. That's why you have this loop here, and this man is responsible for all this cleverness. And I cannot tell more about the internal logic of this system, but he can.
I'm not sure I understand what you mean when you say that what is the RC1 compared to the C1, the shortest and you say what is it rigidly aligned to exactly?
To MNI and to each other. So to common.
Normalized, but it's co-registered.
Co-registered, and I think there are linear deformations like stretching, shearing, all this. I don't know precisely if it is entirely true or not. For sure, there are displacement. They write in sometimes that it is rigidly aligned, sometimes that it is affine co-registration. Definitely, there is nothing nonlinear.
But you cannot do it over the C1 directly.
No, you have to do it on RC1. They are quite specific about this. However, the information about C1 is not lost. The information about C1 is stored in the header of the RC. This is what they encode, because RC is not what you generate. RC is what they give you after the segmentation. And they know what they put in the...
You have the same question, because in our case, we do segmentation without using a template. So basically, you have a Gaussian of everything that is included in the gray matter. So basically, you create peaks, 2 peaks, and then you decide on the threshold where you put it. And then
but this is semi-manual. This is automatic. And they have multi-Gaussians for each these two classes.
And then you decide the threshold.
And they use priors.
Not necessarily.
Here they use priors.
In our case, we don't use priors because we don't have a template.
I don't mean that it is the only way to do it. I just firmly follow the guidelines and the instructions, which are.
But also here, I think the goal is different. Here, you want to compare to, you want to estimate the gray matter and then compare across groups. So I think you are looking for a normal distribution, right? When you do that. So I think the goal is different. when you want to estimate the thickness of the gray matter in a group of subjects that has dementia, or you want to be able to compare control group. So I guess this is also a good reason to use a template and to have a reference. No?
The template doesn't give you an idea of how normal or not normal is your segment at the level of segmentation.
But then this is your reference from which you will be able to compare two groups, right?
The reference is not the template of one group. It is the common template of two groups. You have a group of Alzheimer, a group of healthy controls, and you collapse them and you create a common template. And then you incorporate covariates, age, gender, etc. So you fit them together so that the anatomy is precisely the same, but the deformations are different, obviously. And since the deformations are different, you can do a different modulation of the intensity of the segmented gray matter images. And then you compare between groups those images, and this is what is the estimate of gray matter density, how they call it. Yeah, 6 iterations at least. This is what is proposed by default, and there are a number of parameters to change, but I didn't change anything because I don't understand them. But this is the outcome. At the very end, it gives you this kind of file, and it is an extremely important file for further processing and analysis. Those are flow, nonlinear fields that parameterize the deformation. For each individual image, those fields are different. It is an NII extended file which contains 3 volumes, X, Y, and Z displacement. If you remember, we can go back. You see this black spot. How did it come? It comes obviously from this nonlinear deformation of the original brain. You see, it bumps up here in the occipital cortex. And obviously you have to push it in. And this is what is encoded in this X image. You displace it from left to right, and it is black. So if it is from right to left, it is white. And the same for Y displacement, and the same for Z displacement. And this is what is going to be used for normalization first, and second for modulation. How to run DARTEL? There is no key for DARTEL in the main menu. That's why you already need batch editor. And you go to SPM, Tools, DARTEL Tools, and run DARTEL, create templates. Here you indicate a number of images, always in the same order for different subjects. Gray matter for 1. Block of images, and Y matter for other block of images. And the order must be precisely the same for the two. So first, gray matter corresponds to first white matter, the same individual. Second gray matter, second white matter. 10 gray matter, 10 white matter. same number, same order. I didn't change anything else. And this is what it gives. So the first template, as you see, it is quite unclear, quite smooth, and you see that the anatomy doesn't correspond well in different individuals. But template 6 is much sharper. You can discriminate major and even minor structures, and this is how the ultimate normalized white matter segment will look, plus or moins. And in total, let me remind you that 20 subjects takes an hour on a server. So the next step is normalization. Original template. Here you use actually two templates, the template from the group and the global template. Both of them are essential for spatial normalization. But since the difference between this and the group, the MNI template, is a minor difference, the normalization step takes surprisingly short. This is quite a fast operation. And this gives you a normalized image, but more than getting a normalized entire anatomy, you are interested to get the original gray matter segment to be warped towards the template. So this is an example of original segmentation. All of them are very different, as you can see. And this is an example of warped gray matter images. As you see, they are much more uniform, though still not, they are not perfectly the same. And that's why they do it at the top of it another step of smoothing. So this is a slide dedicated to modulation. As I told you before, the volume is locally enhanced and locally suppressed as a function of the deformation field, which is encoded in this U underscore file. For each subject, one file.
So the deformation going from the gray matter of the subject towards the gray matter that has been of the template.
The local template. Yes, exactly. But at the level of the step of normalization towards MNI, you indicate just this local deformation field, and the rest will be estimated online. So this is how you do it. You go to SPM, Tools, DARTEL Tools, and this time you select Normalize to MNI space. Here you have to indicate few important things. First, you select DARTEL template. By the way, this template, the program drops in the first folder together with the first subject. It will stay there, not separately. But you can find it. It is called template 6. If you indicate 6 iteration, you can do more, but the default is 6. Those are flow fields, the deformation fields, starting with this. Those are 20 files, each for each subject, one for each subject. They are generated at the stage of DARTEL create template. You indicate images. Pay attention that now you select C1, not RC, but C1, which means the native image. Voxel size, bounding box. You can reduce it, you can change it. By default, it is not a number, which means that it is going to be the same as the template. And here you select for preserve, you preserve amount. And preserve volume and preserve amount. In order to get a modulated image, you preserve amount. If, well, for other purposes, you normalize your fMRI data using DARTEL procedure, which is a valid and a recommended manner, you indicate preserve volume because you don't want any modulations there. You just want it to be changed in their geometric position, but not enhanced or suppressed. So this is a scheme of the stages that we pass. Individual anatomy, affine transformed. You use canonical Montreal template. Then you get segmented native gray matter. After you get a normalized anatomy, and this anatomy is normalized towards the MNI template. Then you get normalized grain matter, which is more or less the same color everywhere, but very different in the geometry from the native. And this is the same geometry, but different brightness in different parts as a function of the deformation. This is the normalized modulated. And at the end, don't forget that you also indicate here in the very same procedure for normalized to MNI, you indicate Gaussian smoothing. And this is the kernel of 8 millimeters. They recommend smoothing because they say that otherwise you get aliasing, which is sort of waves over your image because of interaction of different frequencies of sampling between the source and the final result. So what do we show here is the comparison between a smoothed gray matter modulated C1 segment for one individual and the template, the MNI template. As you see, it corresponds very well, though it is more smooth because it is smoothed. At the end, you run statistics. You just do second level. You indicate the directory. Here you have to separate two groups of your images, and you take those results of smoothed, modulated, gray matter segments. It is very much recommended to include a covariate of the total intracerebral volume because it affects very much global deformations. Imagine that two individuals have different size of the brain, and those who has a large brain will have in the modulated image a reduction of volume for all the cerebral areas. The other one will have sort of an expansion of all the areas, which indicates a global trend, but not a local change in the brain. That is why you include a measure of total intracerebral volume for each individual, but you don't include it into analysis. So all the variance that is related to the brain size will be eliminated from your contrast. And at the end, I found that it is better to indicate the explicit mask. Otherwise, it spills all over the head, and this is not what we want. In order to measure the volume of tissues, you can use another utility in SPM batch editor. You go to util and you indicate tissue volumes. And here you can pick this file, which is generated in a regular manner for all the segmentations that you have already done. Doesn't matter if you choose to save the files or not, you always have this SPM8 mat file, and this procedure is much faster than the segmentation procedure. You can do it. If you saved all the volumes, that's fine. You can use this function get totals, which is written by some kind researcher. It is very easy, and it can measure specifically the volume of your image, white matter, gray matter. Then you average them, sum them up, and you get. Thank you. I think that's all. If there are any questions, I will be happy to answer.
I have one question. It's about the procedure that you described. When you include